Let us start. My name is Monika Maleszewska. I'm the organizer of the Bay Area Chromatin and Epigenomics group. And today you're attending our event on machine learning in epigenomics. First of all, I want to say, I hope you can see my screen. Great. First of all, I want to say welcome to everyone. It's really nice to see some familiar names and faces today, but I also wanted to extend a very warm welcome to everyone who is joining us for the first time. Welcome to BASE, and we're thrilled to have you. I like to think that BASE brings together diverse professionals who share the interest in epigenomics, and we do that via scientific, technological and networking events. We meet to learn, to share knowledge, but also to discover and in long term maybe create new opportunities in epigenomic science and industry. We've been active a bit over 10 months and we're a bit over 300 members today. And we typically hold two types of events, a bit larger BASE events like the one you're attending today, which typically consists of a couple of talks and discussion, and these are usually centered around a certain theme, like today's theme of machine learning in epigenomics. And we also, every other month in between, we hold Chromatin coffees, which are more of a social type of event, where we need to learn who we are, what we do, and share some epigenetic news. So I'm just going to very quickly walk you through our agenda today. First, we will hear Sofia's talk, and I will introduce Sofia in just a moment. Then we will have a social breakout session, and you will meet some of our members, Brad, Mihai, Yihui, Data, and Sofia in the breakout rooms. After that, we'll reconvene into the main session to hear our keynote talk today from Anshu Kundaja, and we'll be closing the event close to 7 p.m. I will have one more short announcement for you at the end, so hopefully you can stay till the end. So without further ado, it is my pleasure to introduce our first speaker today, Sofia Kyriazopoulou-Panagiotopoulou. She is the principal scientist at Arsenal Bio, and she will give us the overview of machine learning applications in epigenomics. Sofia, thank you so much for doing that, and the floor is yours.
Thanks, Monica, for the intro. Also, thank you for saying out loud my entire name. Okay, hopefully you can see my slides.
Yes, we can.
Okay, great. So again, thank you all for joining. As Monica just said, I'm currently principal scientist at Arsenal Bio. Necessary disclaimer, I'm not here to represent my employer or any previous employer. Really here to kind of share my excitement about epigenomics and machine learning applications in epigenomics. So yeah, we can get straight to it. Yeah, so this audience probably needs no introduction to epigenomics. However, I'd like to take a moment to highlight that the epigenome encompasses really a variety of mechanisms that affect genome function. First, we have mechanisms that affect how DNA and nucleosomes are packaged into chromatin. Histone modifications, chromatin remodeling complexes, and transcription factors determine how tightly the chromatin is packaged and therefore how accessible it is to the transcriptional machinery. However, the word epigenomics also encompasses additional mechanisms for regulating gene expression in a dynamic and sometimes reversible fashion, such as DNA methylation and the activity of non-coding RNAs. DNA methylation is associated with repression of gene expression, while non-coding RNAs act really in a variety of ways to tune the levels of expression, for example, by repressing translation or inducing RNA degradation. Of course, we cannot understand the epigenome if we cannot measure it, and we definitely cannot apply machine learning if we cannot obtain measurements in a high throughput way. Thankfully, many components of the epigenome can now be quantified using sequencing-based techniques. For example, methods like ChIP-seq use antibodies to enrich for regions of the genome that are attached to DNA binding proteins. And these fragments can then be sequenced and mapped against the genome to reveal the regions where the protein was binding. And the same technique can be used to select for histones that are tagged with specific histone modifications. ATAC-seq is another approach that has recently gained a lot in popularity. And this method allows us to identify regions of the genome that are open or accessible, and to differentiate them from regions that are closed or protected, either because they're bound by DNA binding proteins or because they're wrapped around nucleosomes. And using these methods, we can generate regulatory maps of, for instance, the levels of histone modifications or chromatin accessibility across the entire genome. And over the last decade, researchers have generated these tracks across species, cell types, tissues, and diseases, giving us really very rich data from which we can start to extract patterns. And speaking of patterns, this is exactly what machine learning is designed to do. And indeed, I like to think of machine learning algorithms as pattern extractors. A common example here is a common example of a task is trying to automatically detect whether an email message is spam or not spam. And for this task, we would start with a training data set of labeled examples. And during training, we, or really the computer, will use these labeled examples to learn a discriminatory function. And we do this by finding patterns that characterize spam or non-spam emails. And with this function at hand, we can then make predictions for new unseen examples. Now, some of you might be already familiar with this notion of classification or with the relevant notion of regression, but really machine learning and pattern extraction can do much more than that. So sticking to the realm of natural language, we can take very large amounts of text, for example, all of Wikipedia, and find and learn patterns of human language in an unsupervised way in order to build what is called a language model. And a language model really encompasses grammatical and logical patterns that underlie the English or really any other spoken language. And such language models are really the core of many algorithms that many of us use every day, like auto-completion, machine translation, speech recognition, and synthesis. And a language model is really what allows the computer to know that when, for example, it sees the phrase, "the quick brown fox jumps over the lazy blank," the following word is much more likely to be, say, dog than banana or a verb. These days, there's a lot of talk of deep learning, and deep learning algorithms are just a subset of machine learning algorithms, and they are characterized by their ability to capture very complex patterns. Without going into the details here, they achieve this by using a series of layers that each of them builds on top of the previous layer, and this allows the model to learn kind of increasingly complex patterns of the input. So high throughput NGS-based assays combined with the power of machine learning are helping us answer really fundamental questions regarding epigenomic regulation. In particular, how does the genomic sequence determine epigenomic signals? How do these epigenomic signals relate to disease? And how does the epigenome vary across cells? And in the remaining time, I'm going to highlight just a few examples of how machine learning helps us address these questions. So perhaps the most fundamental question regarding epigenetics is, how does the static genomic sequence determine diverse epigenomic signals across tissues and across cell types. So the central dogma of molecular biology that I'm sure you're all familiar with determines the functional output of the coding parts of the genome. So it is reasonable to ask whether similar rules exist for the non-coding parts of the genome. Using techniques like, for example, ChIP-seq or ATAC-seq, we have long known that transcription factors and other DNA binding proteins have specific sequence preferences, or in other words, they tend to bind to specific sequence motifs. For example, here I'm showing you a so-called sequence logo that depicts the sequence preferences of CTCF, a transcription factor with really crucial roles in genome organization. And here the height of its letter corresponds to the strength of the sequence preference. So a tall letter means that CTCF really prefers that letter or that base to be present at the specific position in order to bind, while shorter bases suggest that there isn't such a strong sequence preference. Similar, we know that even nucleosomes, the most basic unit of chromatin, have weak sequence preferences, preferring, for example, to bind sequences with certain periodic AT patterns. So these and a lot of other similar observations suggest that there exists an underlying grammar that determines the functional output of the non-coding genome. And thinking of the genome as a type of language led to the application of deep learning techniques. So here I like the analogy to machine translation. The goal of machine translation is to take a phrase in one language and translate it to a phrase in another language in an automated way. And these days, this is done using deep neural networks that really translate complex patterns in the source language to equally complex patterns in the target language, using a series of neural network layers. Similarly, a deep learning model can extract complex sequence patterns from the genomic sequence and learn to predict the genome-wide signal of epigenetic marks, such as histone modifications, ATAC-seq, gene expression. So as a more concrete example of this, this is a model called Basenji that was developed by Kelly and colleagues. And the model takes as input genomic sequence and is trained to predict over 4,000 different epigenetic tracks. These tracks include the binding of transcription factors, the levels of epigenetic modifications, DNA accessibility, and gene expression across many different tissue types. The model achieves this by using multiple so-called convolutional layers that are particularly good at extracting motif-like patterns of the input sequence. So here on the right, I'm showing you some predictions for a small region of the genome. And here each pair of tracks, for each pair of tracks, the first track shows the true signal and the second one shows the signal as predicted by the model. And you can, I think, appreciate that the predictions are extremely accurate. Actually, such sequence to function models have proven extremely successful in a variety of tasks. There has already been kind of multiple generations of such models for the prediction of histone modifications, transcription factor binding, and genome accessibility. And these models include the model that I just showed you. Similar models have been employed for the prediction of DNA methylation, 3D genome structure, as well as the identification of targets of non-coding RNAs. And I think it's worth noting here that these deep learning models have also been used to shed light into the coding genome as well. For example, such models can very accurately predict alternative polyadenylation or splicing patterns, and all that just using the genomic sequence as input. So at this point, you might be wondering, what is the use of these models? In the previous example, if we have already performed a ChIP-seq or a ATAC-seq experiment, why do we need a model to predict that signal? And the answer to that is that a model that can predict the functional output of any arbitrary DNA sequence allows us to perform in silico experiments that really are impossible to perform in nature. In particular, we can ask, what will happen if the genomic sequence got mutated? This process is called in silico mutagenesis and has proven really extremely useful in understanding the relationship between the genome and the different gene regulatory signals. And to see how this works, it's worth looking at an example from the same Basenji model that I introduced earlier. So here on the top, we have the CADE signal for two different cell types. And CADE is an assay that measures the activity of transcription start sites. So it's a proxy of gene expression levels. We know that this is the importance of each base of the genome in determining the CADE signal as inferred by the Basenji model. So here, taller bases suggest that this base, or the model thinks that this base is very important for determining kind of the expression output, while the shorter bases are kind of less relevant to gene expression. As you can see, there's a set of bases here in the middle that the model thinks are very important, and this actually matches with the binding motifs of two different transcription factors shown here in the box. You can see that there's a base here that is highlighted in this vertical box where the reference genome does not match the binding motif. So the reference has a G, while the binding motif of the corresponding transcription factor has a slight preference for an A. The heat map on the bottom shows the result of in silico mutagenesis. So it shows the predicted change in the expression signals when every base in the genome is mutated to every alternate base. And as you can see in this highlighted position, the model predicts that an A would lead to increased expression, presumably because it will lead to increased binding of this transcription factor. So this example demonstrates how a powerful model combined with in silico mutagenesis can really shed light into the gene regulatory effects of genetic variants. Apart from helping us understand the regulatory activity of the genome, such models, the ability of these models to be interrogated and to make predictions for really arbitrary genomic sequences opens up new opportunities for synthetic biology. The goal of synthetic biology is to engineer cells with desired properties, and that of course involves engineering both the coding and then uncoding parts of these genomes. Models like the one I just showed you allow us, for example, to design regulatory sequences with specific functions, design genes with desired splicing patterns, or design non-coding RNAs that target genes of interest. So we just saw that machine learning can help us use epigenomic signals to understand the relationship between genotype and phenotype, but can we use the tools of machine learning and epigenomics to actually diagnose disease? Many of you might have heard of cell-free DNA and liquid biopsy for the detection of cancer, but how does this actually work? So cell-free DNA is DNA that circulates in blood, urine, and other bodily fluids. It normally arises from dying cells of hematopoietic lineages. These dying cells essentially shed their DNA, which is quickly degraded in the bloodstream. And as a result, the cell-free DNA consists of very short fragments, and these fragments typically correspond to regions that were protected from degradation, either by being bound around nucleosomes or by being bound by other DNA binding proteins. Now, certain pathological conditions, including cancer, lead to increased amounts of cell-free DNA. So in patients with cancer, the tumor also becomes a source of apoptotic cells that come from the cancerous tissue. There has therefore been increased interest in using liquid biopsy to detect cancer from patients' blood. And to understand how this works, you have to kind of go back to the process that generates cell-free DNA fragments. In a patient with cancer, as I said, the cell-free DNA will come from two different sources, from cancer cells and from kind of normal hematopoietic cells. Due to the quick degradation of cell-free DNA, the ends of cell-free DNA fragments will tend to align with the ends of nucleosomes or DNA-bound proteins. So you can use cell-free DNA to infer essentially DNA structure, which as a result tells you a lot about DNA accessibility and DNA function. So as a more concrete example, this is the pattern of cell-free DNA fragments in a centromeric region. And the fragment endpoints reveal very tightly arranged nucleosomes, which is kind of consistent with the heterochromatin structure of the centromere. While on the bottom, you see a region around a TF binding site, and you can see how the transcription factor is displacing the nucleosomes around it, and this is reflected again in the patterns of the cell-free DNA fragments. So since gene expression differences imply different nucleosome organization, we can use the information about the distribution of cell-free DNA fragment ends to detect changes in gene expression patterns. For example, we can use samples from healthy donors and donors with cancer and train a model that uses the cell-free fragmentation patterns to detect, say, the overexpression of oncogenes. Furthermore, since gene expression and genome structure are highly tissue specific, we can also use the fragmentation patterns to detect the cancer of origin. So although this notion of liquid biopsy is still kind of under very active development, very similar ideas have been very successfully used in non-invasive prenatal testing, or NIPT. So during pregnancy, placental trophoblasts also contribute to the cell-free DNA in the parental bloodstream. And these placental trophoblasts are of fetal origin. So their presence in the bloodstream allows us to detect genetic abnormalities in the fetus using a simple blood test in the mother. And this is the basis of non-invasive prenatal testing. Now, a key step of NIPT algorithms is the estimation of what is called the fetal fraction. So the fraction of the cell-free DNA fragments that originate from the fetus as opposed to the mother. Now, it has been observed that these cell-free DNA fragments of fetal origin tend to be shorter than the parental fragments. In addition, similar to what is happening in the case of cancer, the fragmentation patterns are expected to be different between the trophoblasts of fetal origin and the cell-free DNA from hematopoietic cells of maternal origin. So based on these observations, researchers have built regression models that can accurately infer the fetal fraction from the observed fragmentation patterns across the entire genome. So we've just been talking about the variability of expression patterns across tissues and how we can use this variability to detect disease. But can this leads to the next reasonable question, which is how does this signal vary across cells, even within the same tissue? So the ATAC-seq protocol, which I mentioned earlier, which measures DNA accessibility across the genome and which can reveal a lot of information about genome structure, has been adapted to allow measurements at the single cell level. And one way to do this is using the 10x protocol that encapsulates single nuclei into droplets. However, single cell technologies have created new computational challenges. So a typical Single cell study might measure the DNA accessibility in thousands or even hundreds of thousands of regions across hundreds of thousands or even millions of cells, and potentially across multiple conditions. So, for example, multiple donors. This obviously results in really high dimensional data. It is common practice to try to project this data into a lower dimensional space in order to analyze and visualize them. So that on its own presents a huge computational challenge. Furthermore, the output of these single cell protocols tends to be much more noisy than, say, a bulk ataxic experiment. And this is mostly due to the limited starting material when you're only sequencing individual cells or individual nuclei, as well as to potential errors that arise from the assignment of barcodes to the cells of origin. So the first step of the analysis of such data is always to try to sort of clean it up and remove potential batch effects. And a set of approaches for doing this rely on this notion of autoencoders, deep learning models that are often used in image processing as well as other areas. To understand how an autoencoder works, you can consider the example application of character recognition, where we want to build a model that recognizes handwritten characters, and at the same time is kind of robust to noise. To do this, we build a model that essentially takes a noisy image as an input and learns to reconstruct a cleaned up version of that image. The model consists of a so-called encoder that projects the image into a lower dimensional space, followed by a decoder that takes this lower dimensional space and reconstructs the full image. And this is kind of where the name autoencoder comes from, in that the model really learns to reconstruct its own input. And here, going through this bottleneck of the lower dimensional space is a key component of the model. Because by doing this, the model learns to extract the salient sources of variation while throwing away noise sources of variation. So in the single cell analysis world, such methods have been successful to remove batch effects in order to highlight the biologically relevant sources of variation. And here, this kind of bottleneck layer of the autoencoder model can also be used as a basis for a lower dimensional projection of the input and for 2D visualization. So you kind of get two birds with one stone, both cleaning up the data and removing batch effects, as well as getting a dimensionality reduction of your data. So deep learning, and in particular autoencoder-based methods, have also been used to tackle other challenges that arise from single cell data analysis, such as integration across data modalities, for example, RNA-seq and ATAC-seq, as well as annotation of single cell data with the cell type of origin. These days, it's not uncommon, as I mentioned earlier, for a single study to look at hundreds of thousands or millions of cells. And this obviously results in kind of really big data. And such big data are really commonplace in the deep learning world. So single cell analysis is actively benefiting from rapid improvements in deep learning, such as GPU acceleration and constantly improving open source deep learning algorithms and libraries. So in summary, I presented just a handful of examples that demonstrate how the combination of high throughput epigenomic measurements and machine learning help us understand how the epigenome is established, how it varies across cells, and how we can leverage it to detect disease. Of course, this is by far not an exhaustive list. And as with many other areas of biology and medical research, it's the role of machine learning in this area is expected to increase. And I believe that this is to a large extent driven by the concurrent development and improvement in experimental assays, such as single cell assays. So with that, I'd like to thank you. And if there's time, I'm happy to take questions.
Thank you, Sophia. That was amazing. We have a few questions, so I'm going to scroll up to get to the first one. I think we can take a couple. So first question I received is as follows: epigenome is tissue specific, but primary sequence is more or less the same for all tissues. What other input features are typically used to predict tissue specific epigenetic activity?
Well, that's kind of the magic of it, that there's no other inputs, but these models basically are trained on tissue-specific data, so they learn tissue-specific outputs. For example, the Basenji model that I mentioned earlier is trained on epigenetic tracks across many different tissues, cell types, conditions. So that's where it learns the tissue-specific information from.
Thank you. And another question we have is, for single cell data, how many cells or data points are generally needed to enjoy the benefit from deep learning methods?
I would say like in my experience, any single cell data set that is generated today will benefit from like these deep learning applications or these deep learning algorithms. So in other words, yeah, people don't do like single cell experiments of tiny scales. That wouldn't be, that kind of wouldn't make sense, even like from an economical point of view. So yeah, any practical application these days would benefit from these algorithms.
Great, thank you. We have another question. Autoencoders seem great, but in biology, is there a potential problem of data artifacts being generated by autoencoders when abstracting the raw data?
Yes, there is. Like artifacts, well, yeah, the autoencoder model, it will try, basically, it will remove some sources of variation. And some of these sources of variation will be noise, but it could also remove biological sources of variation. This is almost inevitable in biological applications, in that in almost all practical cases, the sort of your batches correlate with biological sources of variation. So it's almost impossible to not remove some level of biology when you use these methods. And on the other hand, you could get the opposite, where you don't remove all the noise that is present in the data. So yeah, oftentimes you need to sort of tune the models and kind of do a few rounds of, say, parameter tuning to make sure that you have a satisfactory balance between those two, removing too much variance or not removing enough variance.
Great, thank you. I think these are all questions we have here. Maybe, oh no, sorry, we have another question. So let's take maybe one more. Sequence-to-sequence language models currently rely on transformer with attention mechanisms. Are these models being used in epigenetic machine learning applications? It's a seq-to-seq.
Yeah. That is correct. The NLP, the natural language processing world, has been moving kind of away from like simple convolutional networks and more into like these attention-based transformer models. I haven't seen, I think, an application in the epigenomics space necessarily, but there are applications in kind of the biological space. For example, transformer models have been used to model proteins. So yeah, applications kind of the epigenomic, I think it's just a matter of time when we'll soon see papers coming out.
Great. Thank you so much, Sophia. This was great.
Yeah, thank you all.